distinctly sour with a golden crust. Legend has it that miners ran off to look for gold, forgetting all about the half-made bread dough they'd started. Later, they discovered it tasted very different from other batches, sour. They didn't know it, but bacteria would become the flavor of the town. Sourdough bread is made from a special sourdough starter. At Bodine, the same starter, or mother dough, has been used every day since 1849. Willie Joseph has been a baker at Bodine for 33 years.
We put flour, water, mother dough, and we mix for 10 minutes. That's a normal fermented dough. It's normal with flour and water, and the mother dough it's refreshed every day. From a piece of dough, you duplicate it for tomorrow, and that makes the bread sour. If you don't have the mother dough, you don't get no sour.
No one knew what exactly gave sourdough bread its flavor until 1970, when researchers from the United States Department of Agriculture uncovered the secret of the sour, a unique strain of lactobacilli bacteria. They named it, as you might expect, Lactobacillus San Francisco.
For so many years, people produced this product really not understanding what was going on. Now, scientifically, we understand what is actually happening to produce that loaf of bread. The art is now melded with the science.
The steam out of the oven comes and all that flavor, all that smell, you know, people get all, so good. It's everything. Mother dough, San Francisco, the fog, the bacteria, the climate.
A glass of milk, a slice of cheese with your bread? Call on bacteria. If this is a machine for turning grass into milk, its gears are bacteria. Trillions of bacteria are at work in the first of 4 stomachs in a cow. As they do in us, bacteria convert feed into nutrients, which eventually become milk. When the milk is used to make cheese, bacteria play starring roles. Specific bacteria provide the different flavors of cheese and put the holes in Swiss cheese.
We put it into a warm room at about 70 to 74 degrees, at which point the propionibacteria form gas, and the gas accumulates and we have eyes.
Sounds pretty good. A little solid over here.
There we go. Smell that. Nice eyes. Primo.
And then there are the bad bugs. You've probably heard of necrotizing fasciitis. Maybe you know it better as this. Flesh-eating bacteria. The villain is a group A streptococcus, the bacterium behind strep throat. It's commonly found on skin and in throats and usually doesn't cause problems. But another strain can kill with frightening speed. 11 people died in England in 1994 from a type that destroys human tissue at a rate of an inch an hour. Outbreaks of the disease swept through the headlines and popular culture.
Please tell us it's nothing.
He has necrotizing fasciitis from the alligator bite.
Oh, my God. What is this, oh, my God? What is this, oh, my God, fasciitis? Is this what I think it is? Is this the flesh-eating bacteria?
Yes.
Oh, my God.
It infects 1,500 people a year. It may be comforting to note that more people are hit by trains than by this bacterium. Or maybe not.
Now's not the time to panic.
We have a lethal strain of streptococcus from an unknown origin. 3 patients are dead. The health marshal's in my office. When would be a good time to panic?
These are invasive diseases, infections that occur when strep enters the bloodstream, lungs, or muscles. It's the kind of thing that suddenly killed Jim Henson, the creator of the Muppets, in 1990. Who gets these deadly strains of Group A strep and why remains a mystery.
Today, doctors at Children's Hospital stated that one of the children allegedly poisoned by Jack in the Box hamburgers died this morning from complications.
His heart could no longer support his blood pressure.
Researchers know more about the disease caused by E. coli. In 1993, 4 children died and more than 700 people were sickened in the Pacific Northwest after eating undercooked hamburgers from a fast food chain. The cause? A bad cousin of the harmless bacteria living in our intestines.
According to state health officials, there are over 100 cases of E. coli poisoning connected with this case in western Washington alone, and there could be more.
This bad E. coli lives in the intestines of cows. It gets in ground beef if the parts of the cow are not properly cleaned and separated during meat processing. Adequate cooking, however, will kill the organism. Only a small percentage of bacteria may cause disease, but these pathogens have caused untold human misery. Tuberculosis has claimed at least 500 million lives. It is the biggest all-time killer from a single pathogen.
Hello, TB.
Why, hello there, Professor.
Well, there you are. Tell us about yourself.
Sure, I love to talk about myself.
Bacterial diseases have a long and brutal history. One of the most devastating was Black Death, or bubonic plague. The disease, transmitted by fleas, marched across Europe and Asia in the 14th century and killed a third of the population. The death rate from bubonic plague was 90%. With no medicines to fight the bacteria, doctors could do nothing to cure infectious disease. The battle continued until the 20th century, when an accidental discovery changed the course of medical history.
On the return trip, the planes bring wounded back to the hospitals in Britain. Gangrene, from which millions have perished in past wars, has been conquered by the miracle of penicillin. Scientists are manufacturing...
The miracle began in 1928 when a British scientist named Alexander Fleming went on vacation. He didn't bother to clean up first, just placed his bacterial cultures in the sink. When he returned, he noticed one dish had gotten moldy. The mold killed the bacteria, but Fleming couldn't stabilize or purify it. That took a 4-man team from Oxford and 10 years. They named the drug after the mold. Penicillium led to penicillin.
Thousands of men, thanks to penicillin and plasma, will come home to their thankful families. A whole world of peace to come will reap the benefits of this great wartime medical discovery. Science has...
Penicillin was the dawn of a new age in medicine. At last, doctors had the power to heal. It seemed man had infectious disease on the run.
In the 40s, when antibiotics really came into general use, there was such a kind of high that developed because we were taking things like streptococcal blood poisoning and pneumonias, gonorrhea, syphilis, and all of these things were being cured. It was a miracle.
4-year-old Elijah was a sick little boy, and his mother Vivian was worried.
He wouldn't eat, he wouldn't play, he wouldn't do anything. He'd just lay there. He said his foot was hurting him. I asked him, did he step on something or did he hurt his foot? And he said no. It was very unusual. And after his fever wouldn't go down, I knew it was serious.
As an experienced mother of 5, Vivian had never seen an illness like this. After 2 days of high fever, she brought Elijah to the hospital.
Her when I go like that? Does it hurt when I go like that?
Vivian learned Elijah's fever and pain were caused by a bacterial infection in his foot. The bacterium, called pneumococcus, usually causes ear infections, but in Elijah's case, it got in his bloodstream and lodged in the bone. The doctors prescribed antibiotics and expected Elijah to feel better in a couple of days. But 5 days later, Elijah's fever had not gone down. The antibiotics were not working.
In the late 70s, we produced in the pharmaceutical industry antibiotics active against resistant organisms in 3 or 4 different classes. In the early 80s, these products all came to the market, and everybody, all physicians said, well, now we've got something active against all the resistant organisms. And the message that came back to the pharmaceutical industry from the medical community was, in fact, that they really didn't need any more new antibiotics. I think that that was a certain segment of the medical community who didn't understand bacteria and understand how bacteria change and modify and adapt.
This is one way bacteria adapt. A bacterium with a plasmid, plasmids hold genetic information, will connect with another bacterium. A copy of the DNA is transferred. The new DNA may dictate a new food or energy source or tell how to dodge an antibiotic. This sharing is passed on and on to other bacteria, even among different species, and it can happen in less than an hour.
You could say that, well, if organisms really began to exchange information, then all bacteria would kind of be the same. They'd be in an amalgam, but they're not. Each one is kind of very individualistic in terms of what they do and how they survive, and each one has a gimmick in order to survive, so that it has its place, as it were, in the world.
That's little comfort for Elijah and his mother, as the doctors try to find out why he is not getting better. The heavy weapons of medicine are brought out to pinpoint his bone infection. Doctors collect bacteria from his foot in the operating room, then grow it in the laboratory to find out why the medicine isn't working.
It's pretty much where the needle was. Right. We'll see more when we get the pinhole views. Looks like we're on the right track.
Pneumococcus is the leading bacterial cause of pneumonia, ear infections, and meningitis. Throughout the country, the number of resistant pneumococcus strains is rising, and the strains are resisting more classes of antibiotics.
Antibiotic resistance means that the bacteria have developed a way to avoid the killing action of antibiotics, which simply means that if you have an infection and you're given one of these antibiotics, it probably won't work for you.
One way to study antibiotic resistance is to go where antibiotic use is highest. In Wisconsin, nurses Priscilla Goltz and Jane Stofflet are collecting samples of, well, the material produced by runny noses. It's part of a study for the Centers for Disease Control. The rate of resistant pneumococcus in some Wisconsin daycare settings is over 40%.
Okay, and I'm going to give you a bear hug, and you give Tom a bear hug, okay?
Jane's just
and we're
going
just going to check your nose.
Give you a hug, and we're just going to check your nose, honey, okay? What I want you to do is just count to 3, honey, okay? 1, 2, 3. That's all,
done.
Sweetheart.
I'm
That's
done.
all.
Oh,
good
job.
girl.
Good
You
job.
did good, honey. Thank you.
Now you got to pick out a toy.
Good work.
Let's go pick out a toy.
The nurses will return in a year and retest the children to monitor the trend.
Oh, yes.
Daycare centers are often seen as one of the wonderful breeding grounds of new and novel resistant organisms. In many large daycare centers, there may be as many as 30 or 40% of children on an antibiotic at any given time. And so that an organism that is a good colonizer has a tremendous opportunity in a daycare center to colonize lots of children and eventually to cause disease, particularly if it's resistant to 1 or 2 or 3 antibiotics.
Millions of children take antibiotics for ear infections or to prevent them. If the medicine doesn't kill all the bacteria, the infection returns and more antibiotics are prescribed. Doctors are concerned about the overuse of drugs, but don't see many alternatives.
You know, we've talked about the bacteria called the pneumococcus bacteria.
Right.
That is probably the most common bacterial cause of ear infections.
Mm-hmm.
And that's the one that's showing more signs of not responding well to antibiotics.
Oh, okay.
Hi. Look
still has a lot of fluid. It's going to take a while for the fluid that's behind the drum to resorb or go away.
Right.
So with the way his eardrums are looking, I think we ought to put him on a preventative antibiotic.
Okay.
And we'll just do that for at least 4 to 6 weeks.
Okay.
When they're real uncomfortable and obviously bothered,
Right.
I think it's hard to just let that go.
Especially from a parent.
And if he does develop a rash.
Antibiotics are also widely prescribed even when they won't help. 18 million prescriptions are written each year for colds, which are caused by viruses.
Very good.
The sheer tonnage of antibiotics that is used in the world every year, I think, contributes to the problem of resistant bacteria in the community, which then becomes a problem in the hospital, which then becomes a problem in extended care facilities, and they're all inextricably linked.
During the 1950s, animals started getting antibiotics too. The drugs not only curbed disease, but for reasons still not understood, they promoted growth. Today, half of all antibiotics used in the United States are used in animal feed. This little piggy will be fed penicillin and tetracycline before it goes to market. Even if you're a vegetarian, you get a dose. Antibiotics are also sprayed on fruits and vegetables.
I can't think of a microbiologist that would say that the wide application of antibiotics will do anything but select for antibiotic-resistant organisms. And so that's a consequence. So if there are economic reasons for doing it, that may be what the public wants, but they also have to pay the consequences, which will be that those antibiotics may not be useful in the treatment of diseases at some time in the fairly near future.
The lab has reported back to us on the second sample. It showed to be very resistant to the antibiotic he was on. Not only is it resistant to that antibiotic, it actually happens to be resistant to 2 of the other antibiotics we might use. Fortunately, though, we've got one left. It's an antibiotic called vancomycin.
You ready for this?
Vancomycin is considered the antibiotic of last resort. It is very potent, it's expensive, and it can be toxic. Elijah has to be given this medicine intravenously, and he will need to keep the IV in for 5 weeks, even after he goes home. A drastic change from pneumococcal infections of the past, when a teaspoon of pink medicine twice a day did the trick.
There is a feeling, a deep concern about the availability of antibiotics to treat some of the resistant organisms. For some of these major problems, there really is right now only one antibiotic that works. So there's a fear that if the resistance would develop to that particular antibiotic, then we would have a major problem on our hands.
Staphylococcus aureus is the #1 cause of hospital infections. In recent years, the increasing number of resistant strains has caused alarm.
Among the Staph aureus strains in hospitals, many of them are resistant to everything except vancomycin. And so if vancomycin resistance moved into these strains, we could have untreatable strains of a very common resistant organism. That's a fear. Our hospital wards in the 1990s would look like the hospital wards in the 1930s, which was the pre-antibiotic era. There we had people dying of diseases such as typhoid and tuberculosis and diphtheria and pneumonia, and we really didn't have much to give them in the way of therapy.
The sun shines in Colorado almost every day in the year.
It has happened. A patient in a Tokyo hospital developed a vancomycin-resistant staph infection. If the strain travels to the United States, as resistant strains have in the past, doctors will have very few options.
One of the factors that has led to a global increase in resistance among bacteria is the fact that we have very mobile populations of people. You have international travel where you can literally take a resistant organism and transport it from one continent to the other in less than a day. So it's a major problem around the world.
Some resistant strains of disease are untreatable and potentially deadly. In 1994, a Korean woman flew from Baltimore to Chicago to Honolulu. Unknown to her, she had an active case of multi-drug resistant tuberculosis. After the woman died from the disease a month later, the passengers on the flights with her were tested. 15 tested positive for TB. These people may or may not develop active tuberculosis.
If we learned anything from the last 50 years of the antibiotic era, this race is going to continue. It may accelerate because we'll have new antibiotics as well as new classes of antibiotics in the future. But I think we'd be naive to think that just because we have a new class of antibiotics that's active against a currently resistant microorganism, that this process is going to stop. I don't believe that it will.
With the vancomycin, we need to worry about things like the renal toxicity and the ototoxicity, and that's the real problem with the organisms that cause resistance.
So we have a long haul ahead of us. It's going to be going home on IV antibiotics. Essentially, we have very few options to offer him.
You didn't even stop to say hi, did you? Yeah.
Elijah is ready to go home. A nurse will visit him there to set up his IV. He will be monitored carefully to make sure the antibiotic is not damaging his kidneys. 6 weeks later, Elijah's infection is finally gone. This part of South Dakota and the city of Lead is mining territory. People from all over the Black Hills have worked for the Homestake Mining Company, the longest continuously running gold mine in the world. It's a one-company town. Without Homestake, there would be no Lead.
A lot of the miners are like third and fourth generation. Their grandparents worked in the mine, and then their parents. And so it's like a big family.
If you look at this table here, it's nothing more than an automated gold pan. It's doing the same thing the old-timers did in the stream, except it's much more efficient and it's doing a lot faster and a lot more material. See that little golden streak coming down the table? That's the gold.
But that streak is only half the gold in this ore. To extract the rest, the rubble goes to giant tanks of cyanide.
Cyanide dissolves gold just like sugar dissolves in water. It makes a pure solution out of it.
After the gold was extracted, the cyanide solution was dumped into the town's Whitewood Creek, killing everything in the water. For over 100 years, the creek ran black. The Environmental Protection Agency gave Homestake 1 year to develop a plan to get the cyanide out of the creek. The mine managers weren't sure how to tackle the problem and keep the mine profitable. For the people in Lead, South Dakota, profits for Homestake meant jobs for the town.
There's very few things besides cyanide that will dissolve gold, and they're a lot nastier than cyanide.
Homestake's chief executive officer added a challenge: make the stream pure enough for trout. Homestake hired a local biochemist, Jim Whitlock. Whitlock and his team decided to use their background in microbiology to try something new.
We started with a simple knowledge that bacteria could tolerate cyanide, and we knew that some bacteria could tolerate a lot more cyanide than others. In fact, this was a standard means for microbiologists to identify bacteria based on how much cyanide they could tolerate. And so we looked into that in depth, and what we found out is that they actually broke the cyanide molecule into 2 parts, and so that they could actually use it as a food source. This was the only thing that we found that had any potential to work, so it was a one-shot deal, and it had to work. That was a little bit on the scary part.
It worked. Today, all the water from the underground mine is filtered through these tanks, each containing about 20,000 pounds of Pseudomonas bacteria attached to plastic discs.
Now, the bacteria like to attach to things, and they have a little tail that they use to propel themselves around in the water. They'll discard the tail and secrete a slime layer, which is how they attach to the plastic disc. So as the disc rotates very slowly, about 1.5 revolutions per minute, partially submerged in the water tank, the bacteria are attached to the disc. So they're in the water, and then they're back in the air, and then they're in the water. So when they're in the air, this helps satisfy their oxygen requirement. And when they're in the water, then they're actually picking up cyanide and absorbing metals.
Every day, 4 million gallons of clean water pours out of the treatment plant and into the Whitewood Creek.
Once the plant went online in 1984, within 6 months there were fish moving into the stream. Fish, of course, are the best chemists of all. You can't fool them. It's either okay for them or it isn't.
Meet the canaries of the Homestake Gold Mine. Before the water goes through the pipe and into the creek, trout kept at the treatment plant give the final approval. The fish were happy. The Pseudomonas had plenty to eat as they broke down the cyanide for food. The bookkeepers were happy too.
We were looking at a chemical process that would probably have cost us $3 to $4 million a year for chemicals and operation, and we ended up with a biological process that cost us approximately $500,000 a year to operate.
Today, the ore blasted out of the earth provides work for 1,000 local residents and uncounted trillions of bacteria. The result is 1,000 bars of gold a year.
For me to come back home where I grew up and be part of a team that cleaned up an area and completely changed an area, it was very rewarding, particularly because it was at home.
Come back and have a breath test today and see whether or not your antibiotics have worked.
1 in 10 adults has ulcer disease, and it's not stress-related. Now we know the true cause. Helicobacter pylori, a spiral-shaped, slow-acting bacterium that thrives in the stomach. Simple tests will detect H. pylori, and antibiotic treatment can begin.
Very good. We have our first sample.
I think it's an incredible discovery. I mean, that we've actually been able to come so far in where we are with ulcer disease, because for so long it was thought that you needed to get rid of the acid. So the surgery was taking out the acid part of the stomach, making it so that the acid decreased, and also taking out that area where the ulcer is formed primarily. But now we know that it's just a bacterial disease, and all you have to do is just take a 7-day or 10-day course of antibiotics and it's over. It's great.
If even one of these diseases proves to be caused by bacteria, and thus goes from the incurable or where the cure is a very radical and unacceptable to many patients, take it off that list and put it on the list of diseases that can be readily cured by antibiotics, that would be a major contribution.
This is the Institute for Genomic Research, or TIGER. Here they pull out a bacterium's genetic blueprint, its DNA, and break it into chunks that a supercomputer pieces together. If the whole DNA is the book describing a bacterium, the genes are the pages. The computers put the pages in the right order. Once the genetic blueprints of microorganisms are completed, the functions of the genes are studied. As researchers understand which genes are responsible for growth or disease, they can be manipulated for many purposes. And it's given us insight into a much larger question.
We like to think that we're highly evolved, and therefore our chromosomes are probably highly evolved. And bacteria are primitive. Well, in fact, the opposite is true. The bacterial chromosomes are very highly evolved. If you think of organisms that replicate every 20 minutes to an hour, and they've been doing that for billions of years, they've gotten very efficient at everything they do. So there's no wasted letters in their alphabet. Essentially, they're wall-to-wall genes for all practical purposes.
Understanding the genes of microorganisms also opens up thousands of new targets for antibiotics, vaccines, and industrial applications.
It's amazing the diversity that's out there. We know a tiny fraction of 1% of what's out there. And the smartest thing we could do in this research is, go try and find new ways to culture thousands and thousands of new microorganisms and find out their genetic code and find out what's there. It's going to revolutionize virtually everything we do.
Yellowstone National Park is one of the richest places on Earth for all microbial life. Bacteria thrive in the extreme conditions found in the pools. Tourists see the spectacular scenery. researchers come to understand how life can survive.
Many of the organisms that live in these pools have to eat. So those are called heterotrophs, so they have to consume things in their environment. And in order for a microbe to consume something in its environment, it has to make an enzyme that can break that particular item down. Well, if we see a piece of wood in this hot pool and we pull it out and we see that the outside of that piece of wood is bleached white, you automatically ask the question, how did that happen? So when we look at the microbes associated with that wood, studies have shown that these microbes have that capacity because we've cloned the genes from these organisms that encode those particular enzymes and have found that in fact they are adapted to breaking down these wood fibers under these extreme conditions.
By cloning the genes that produce whitening enzymes, they can turn it out in quantity. That means paper companies can use bacterial enzymes to whiten paper and lessen the need for chlorine. It's good for the environment, and it's big business. Enzyme production is an $11.5 billion a year industry. Most antibiotics come from bacteria living in the soil. As the search for new antibiotics continues, scientists are learning just how little we know about the microbial life beneath our feet.
To microbiologists, the earth is the richest source of new biology that there is because there's far more that we haven't studied than that we have already discovered. In a teaspoonful of soil, there are at least 1 billion bacteria that we can grow in culture. But there are many, many more bacteria, perhaps 100 times as many, that can't grow on our culture media. And in fact, we've probably been studying only a tiny portion of the organisms that actually live in soil based on their ability to grow on our Petri plates.
But if the bacteria won't grow in the lab, how do you find out what they can do? How do you test them? Start with the soil.
One of the things we've been doing is extracting the genes from the bacteria directly without culturing them first, just pulling their DNA out directly from the soil and then sequencing pieces of that DNA to describe the organisms that are in that soil. And using that methodology, we find that most of the organisms in the soil have not yet been described.
The search for new microbial life has taken researchers from backyard soil to the ocean floor to the heavens. A meteorite from Mars has fossilized structures that look suspiciously like small bacteria. Scientists are coming to the conclusion that life may be far more common in the universe than we had supposed.
Anywhere that there is liquid water, there is a possibility of life, and we know that here on Earth that's just a fundamental requirement. And in fact, anywhere on Earth where there is liquid water, you find there is life. And so the fact that on planets such as on Mars, where you might have hydrothermal features under some of the ice flows that might make liquid water is very exciting.
The experts say this is the beginning of the golden age of microbiology. Industry, medicine, the definition of life itself are all being changed by single-celled creatures you and I can't even see.
Everyone should look at pond water at least a few times a year if you're a microbiologist, because now the days of technology and DNA and high-tech things, it is nice to go look in a microscope every once in a while and look at that life and that diversity of life, because you can't just put it in a vacuum or look at it as a bunch of A's, T's, G's, and C's and a chromosome. This is not life in itself. So you should go look at pond water. Everyone should look at pond water, and they'll understand in that instant is more about a feeling about what life is than almost any other kind of experience they could have.
Hi there, Lee Murnick. Welcome to our lovely home. Good to meet you. Stacy shook your hand. Right. And look at it's all over your hands.
Oh my God.
This shows how germs are transmitted from hand to hand, person to person, and from surfaces.
Okay, reach for the bottle now. This is the same bottle I'm going to be using for months in the kitchen.
Right.
I'm putting this on my Italian bread tomorrow, making just tasting. And there's the same bottle, cross-contaminated. We now rub it. Oh, yes, we got to rub a little bit of this on. And I reach up. You want a little salt on there? Because you want to touch my salt.
I thought cross-contamination was a good thing. Then I remembered it was cross-pollination.
Cross-pollination is what you're talking about, friend. Where's my whistle? Hello.
That was good. This is bad.
I could have made that line up better, friend. Would you stick around the rest of the afternoon like I happen to be?
This team of Army medics is rehearsing, not for the outbreak of war, but the outbreak of virus. It's a battle where shaking hands can be more lethal than dodging bullets, where the adversary numbers in the billions, where nothing's constant but the body count. Viruses are things on the very edge of life. We can't taste them, smell them, or see them, but they're there. In the air, in the water, in every living thing on the planet. They're the tiniest, simplest form of life on Earth. Yet they know ways to enter our bodies, kidnap our cells, outwit our immune defenses. Viruses are old. Some believe they're older than all other forms of life. And they are patient. Without a living cell to invade, they wait in a crystalline form somewhere between life and death. Some can wait for centuries. Their only goal is to reproduce. Viruses are pure purpose.
Viruses, when they're out there in the environment, are basically just pieces of genetic information wrapped up in a protein coat or some other little bit of protection, vulnerable to all the elements in general, and just waiting for the next living thing that they can grow in to come along so that they can infect and propagate themselves.
It's the form of life that cannot subsist. It doesn't have the machinery with which to live. It can only do so by entering and utilizing another cell for that purpose.
And when it uses human cells for that purpose, we usually end up the losers. Diseases caused by viruses are as potent a force in the history of man as war and natural disasters. Influenza. 15th century Italians called it that because they believed it was caused by the influence of stars and planets. Smallpox was first reported in Chinese literature 3,000 years ago, now extinct. There are 7 different kinds of herpes. Chickenpox is one. Odds are you have herpes right now. Kuru is found only in cannibal tribes in New Guinea, transmitted by eating human brains. Each cold you get may feel the same, but it's not. There are hundreds of different cold viruses. In your lifetime, at least 50 of them will catch you. And the list goes on, as common as colds, as deadly as AIDS.
Viruses are a cause of a lot of misery. There's the viruses you catch in the office, the daycare center, that your kids bring home from school that cause minor disease. We don't diagnose viruses specifically, so that we often pass them off and we don't pay the kind of attention to them that we might.
Viruses have always been difficult to pin down. Even the most powerful optical microscopes could only magnify objects 2,000 times. They revealed a watery world of bacteria and single-celled creatures, eating, moving, multiplying. What they couldn't see was the phantom that destroyed cells with stunning efficiency. Then, in the 1930s, German scientists invented the electron microscope and could finally see a virus at 7,000 times magnification. Instead of bouncing clumsy light waves off objects, it used tiny electrons to magnify. One look through the electron microscope instantly changed how people defined the boundaries of life. They discovered organisms so small, billions and billions of them could fit into a single pixel on your television screen. Scanned by electrons, viruses were revealed to be spheres and spindles and geometric shapes. They looked too symmetrical to be alive. On today's supercomputer screens, viruses look like ornaments from a Martian Christmas tree. This is the AIDS virus. Its core contains the genetic information necessary for the virus to duplicate itself.
It's a package of nucleic acid, of genetic material, enveloped in a set of proteins that allow this virus to utilize a cell for its own replication, because it cannot function independently, it cannot multiply independently.
The genes are protected by a thin shell. The shell is studded with molecules that enable it to dock with the target cell and work its way inside.
There are certain receptor mechanisms, they come together, they attach, they don't attack, they attach, they're incorporated, and they use the cellular mechanism for its own multiplication. It acts as a parasite. It uses these organized systems for its own survival.
The target cell doesn't have to be human or even animal. The tulip break virus has been used since the 16th century to cause color variation in ornamental tulips. Nothing is too small for a virus.
So, naturalists observe a flea, and smaller fleas that on him prey. And these have smaller fleas to bite them, and so proceed ad infinitum, infinitum, infinitum.
Yup, even germs can get germs. An example, the T4 bacteriophage, a virus that attacks bacteria. It lands, hooks on, and pushes its DNA through the cell membrane. Once inside, the viral DNA hijacks the cell and forces it to make more viruses. Within minutes, the swollen cell explodes, releasing hundreds of new viruses to infect other cells. In ancient times, people didn't know about viruses, but they did know this. Getting sick was the last thing most of them ever did. Where did these diseases come from? Was it the wrath of the gods? Omens? Sorcery? The Great Red Plague that struck during the Peloponnesian Wars in the 5th century BC may have been typhus or scarlet fever, or it could have been measles. Smallpox was the scourge of the Middle Ages, killing many and leaving survivors permanently disfigured. When 20th century archaeologists opened Egyptian tombs, they found references to what could have been polio. But diseases of the past were limited in time and space. The world had not yet experienced viral epidemics on a global scale. One of the worst natural disasters in recorded history was caused by a virus. The influenza pandemic of 1918 struck every major U.S. city, dropping people where they stood.
There were at least 20 million people killed worldwide, and it's probably the worst pandemic the world has ever known. There were more people killed from influenza than there were from the First World War.
The war itself was the problem. If there had been no war, that year's strain would have stayed isolated. But when Allied troops met in northern France to exchange fire with the enemy, they also exchanged a lethal strain of flu. So it began.
It then went from France to the United States, and it disseminated from Boston.
At Fort Devens, Massachusetts, one case of flu turned into more than 6,000 cases between September 1st and September 18th. It took only 17 days. India lost nearly 4% of its population. Alaska, 8%. In the South Seas, the death rate was 20%.
People were absolutely panicked because somebody could be playing bridge one night and they would be dead the next day. And so they did all kinds of things. They were wearing face masks, and in fact, the police all wore face masks. They were carrying little pouches of garlic and onion, just anything that they thought would protect them from the flu.
They had no idea that they were leaving exposed the very place where flu enters the body, their noses.
Nothing was known about how to cope with it. And that is something that began to emerge when we got into the 1930s, 1940s, and 1950s. By that time, there was enough knowledge and experience and the development of techniques to make it possible to proceed.
Never before in history have doctors been able to do so much to preserve your health as they can today. And almost everywhere in medicine, the parade of advances that will benefit countless millions has just begun.
Vaccines became a scientist's dream. They promised to rid the world of viral disease. Curiously, the word vaccine comes from a cow disease called vaccinia. In the 18th century, the beauty of milkmaids was a cliché. They alone seemed to be spared from smallpox. An English country doctor named Edward Jenner observed that a surprising number of milkmaids caught a mild, measles-like illness called cowpox, known officially as vaccinia. Jenner extracted the virus from blisters and infected a child, his own infant son. Then, he inoculated the boy with live smallpox virus. It was shockingly risky, but it worked. The initial cowpox injection did cause mild sickness, but it was enough like smallpox that his body eventually built an immunity to both diseases.
We have evolved our own defense mechanisms over the years. We know this is the immune system, which is a very complex system containing many different kinds of cells and functions, whose major purpose, by and large, is to recognize what's foreign and attempt to rub it out.
The immune system has three major functions, which are working in your body right now at this very second. Antibodies are proteins that bind to the surface of a virus, flagging it for death. The body's white cells move in to destroy virus-infected cells. Once you have been infected with a virus, memory cells will immediately recognize it if it enters your body again. This is why vaccines are so effective. the body remembers what the fake virus looks like, so it can protect you when the real virus comes along. One of the most common ways to make a vaccine is to grow it in an unnatural host. Chicken embryos are used to grow human viruses like yellow fever, measles, mumps, and influenza.
Influenza has been grown in eggs for many, many years. It's our standard way of producing virus. It can also be grown in tissue culture and grows very well in tissue culture, but you can't produce enough tissue culture to make large doses required for industrial use.
Every year, flu virus is injected into eggs. After several transfers from egg to egg, the virus will have become better adapted to growing in chicken cells than human cells. When injected into a human, it looks enough like the enemy to cause immunity, but isn't strong enough to cause sickness. The flu virus has found a way around the immune system by mutating, changing the way it looks. Sometimes it changes only slightly. Other years, the change is radical and we are defenseless.
Sometimes you can get partial immunity, which will give you some protection. But when we have what we call a shift, that is usually what occurs when we have major epidemics. Then the immune system doesn't recognize the flu at all. Bears no resemblance to anything the antibodies have seen before.
How much a virus can change depends on how it's made. If it has genetic material arranged in the familiar double helix DNA, then it's pretty stable from generation to generation. In reproduction, when a DNA strand unzips to become two DNA strands, a kind of molecular spelling checker watches the new molecules form to make sure the two copies are identical.
The DNA viruses have a nice copy editing system, so they will correct their own errors, which means that the frequency of mutation is low. The RNA viruses do not have those. And this explains why RNA viruses are so variable. These viruses change all the time.
Which is why the vaccine for a DNA virus like smallpox works decade after decade, while every year we need a new vaccine for RNA viruses like influenza. This frozen library is Dr. Herlocker's collection of flu strains that have evolved over the years. These viruses are not dead, just dormant. Each vial represents a different year, a different strain. If you warmed up and breathed flu vintage 1980, you'd probably get sick. Some vintages of flu are spontaneous mutations. Other epidemics can be traced back to ducks and pigs. They end up with names like swine flu, or the Hong Kong flu, or the Asian flu, because they often come from countries where people live close to their farm animals. Influenza has the added advantage of crossing from one species to the next. Each time this happens, the strain changes. And that means a lot of work for scientists at the World Health Organization and Centers for Disease Control in Atlanta. Every year, it's a race against time.
CDC Influenza Surveillance Laboratory.
Information is gathered from flu watch centers around the world and used to predict what new strain is coming our way. Once this is determined, a new vaccine is developed, hopefully in time. It's hard for us to imagine a world without vaccines for diseases like influenza, smallpox, or polio. In the first half of this century, people panicked when children came down with fever and muscle aches. It might lead to nothing. It might end up with the child spending the rest of his days in a pressurized cylinder everyone called the iron lung.
In my own lifetime, I was aware of the effect of viruses. In 1916, for example, which was 2 years after I was born, there was this severe polio epidemic in the United States, particularly in New York City. And then again, in 1918, 1919, there was a severe influenza epidemic. And in both instances, the effect was quite devastating. Now, I, of course, was too young to understand the significance of that, but I do remember the crippled bodies and the coffins.
Nobody knew why polio caused paralysis or death, much less that it could be transmitted in contaminated food, water, or spittle. Home remedies and preventatives were born out of desperation. When polio struck Franklin Roosevelt after he had gone swimming in a chilly lake, people speculated that the cold water must have been the cause. By the late 1940s, a young Jonas Salk was well on his way to becoming a hero to parents worldwide.
My whole life has been devoted to being a physician, and this is the way in which I chose to practice medicine. And so this is what the pattern of my life has been, to try to either prevent disease or heal or cure in some way.
His breakthrough came when Harvard scientists found a way to grow polio virus in quantity. Salk had the raw material he needed to develop a vaccine.
My whole approach to this, that one could immunize against a virus disease without experiencing infection. Until then, it was believed that you had to experience infection, as in the case of smallpox, rabies, and an attenuated form of the virus.
In his lab at the University of Pittsburgh, Salk killed polio virus with formaldehyde to prevent it from causing disease. In 1955, after first trying it on himself, he began inoculating children. The dead virus alerted their immune systems without making them sick. It was treated as the biggest news in medical history. In the United States alone, 2 generations have grown up without knowing the anguish of the disease.
It was all in a day's work, I like to say. And it was appreciated, especially by your parents, for whom fear was lifted.
Back in 1935, a bacteriologist called the battle against disease one of the last genuine adventures in the world.
The dragons are all dead, and the lance grows rusty in the chimney corner. About the only sporting proposition that remains is the war against those ferocious little fellow creatures, which lurk in the dark corners and stalk us in the bodies of rats, which fly and crawl with the insects, and waylay us in our food and drink, and even in our love.
Today, new crusaders struggle to slay the littlest dragons that lurk in the world of poverty. The rotavirus, a tiny waterborne microbe that kills over 5 million children a year. 5 million, mainly in countries of the developing world. In the war against rotavirus, this is the battlefield, a village in central Brazil. The town is called Terra Firma, but it's built precariously over the Guana River. The Guana River is the only running water here. It's the local sewage system. Unfortunately, it's also the local water supply. In collaboration with the World Health Organization, Dr. Alexander Linares and his team from the Instituto Evandro Chagas in Brazil track the rotavirus, and try to help young mothers battle the disease. But fuel to boil water is hard to come by. Sanitation is hard in a floating slum in the tropics.
The virus is so stable that human-to-human transmission perpetuates in the community. We know that improving sanitation conditions, improving hygiene is very important, but it's not sufficient in order to control rotavirus diarrhea. It is necessary to have an effective vaccine.
The researchers monitor the children's health and test their blood to study how they develop antibodies to the virus. They conduct field trials evaluating the efficiency of a candidate vaccine. This vaccine, if approved, could potentially save 800,000 young lives a year. It would require mass immunization, but that's an area where the World Health Organization already has a lot of experience. In 1967, they decided to kill a disease, smallpox.
We have never driven any infection to extinction. Smallpox was the very first time that finally the world got together and decided that if we were going to cause species to go extinct, let's do it for a good purpose, for a change.
International teams of doctors and nurses were sent door to door tracing the last case of smallpox to Somalia.
And it's absolutely amazing to see the way they did it. They had groups, teams of doctors and nurses, and go all around the world in the deserts, in the Sierras, and all around every country in the world. They vaccinated everybody.
The smallpox virus can only grow in human beings. Vaccinations kept it from spreading to new hosts, so when the last victim recovered, the chain of transmission was broken forever. Smallpox, the disease, disappeared from the earth in 1978. Smallpox, the virus, is still around. These stark hallways lead to what may be its final resting place. Only 2 collections are left in the world, and they are kept in Moscow and here at the Centers for Disease Control in Atlanta. The World Health Organization wants them destroyed, but not everyone agrees.
We didn't want to totally eliminate the virus stocks because we didn't really understand the virus and what made it tick. It's part of the whole basic fundamental argument about diversity, biological diversity. Can we afford to lose these organisms that have established themselves in a particular ecological niche?
But the greater danger is the possibility of smallpox accidentally escaping from a freezer somewhere where there might be a forgotten stock stored, or years from now where after people have forgotten about it.
The whole idea of smallpox escaping and decimating unimmunized populations may seem like science fiction, but it happened hundreds of years ago. In 1519, Hernando Cortés invaded Mexico and conquered millions of Aztecs. He did it with less than 600 men, a few horses, some primitive guns, and, with a weapon he didn't know he had, a virus unknown in the New World. The Aztecs and their immune systems were defenseless. What was often attributed to the will of God was actually the work of man. This time, the transmission of the virus may not have been so innocent. According to some experts, Lord Jeffrey Amherst of Massachusetts Bay Colony ordered the distribution of smallpox-infected blankets among local Indian tribes. The age of exploration became a perfect vehicle for viruses. Today, it's no different. just more efficient. Global commerce, overpopulation, teeming cities. Progress. But progress is not without its price. We continue to create highways for what scientists now call viral emergence, a steady stream of old viruses getting out to look for new hosts.
As people introduce themselves into environments that used to be inaccessible to human habitation and to human exploration, suddenly these viruses spring out, and they spring out at us because we're putting ourselves there, and we're putting ourselves in their path. And so in many cases, these viruses that seem to come out of nowhere really do come from somewhere. They come from nature. They're just like all other life forms. They've been there. And what's new is the opportunities we have given them, without realizing it, to meet new people.
Among the planet's remaining natural environments, few are changing faster than the tropical rainforests. Experts estimate that 90% of the 14 million species on the Earth are unknown to us. And all of those unknown plants and animals can harbor unknown viruses. That's what we face when we destroy the rainforest.
I think we're going to see emergence of more disease, and I think everybody would agree we need people out there who are familiar with the diseases in local areas, who can describe the trends in diseases, and who will see a new disease when it comes along.
Everybody's got the fever. That is something you all know.
Senor Helio Augusto Cardoso Sareva is part of a team of physicians and scientists trying to predict the pathways of viral emergence. His job? Bait. Augusto is bug bait. He waits patiently in the canopy of the Brazilian rainforest for mosquitoes to bite him. Augusto and his colleagues are part of an early warning system for arboviruses. Arboviruses are carried by insects that live in the treetops and on the forest floor. The bugs come to a sudden end as they and their cargo of viruses are frozen in liquid nitrogen for the 10-mile trip to the Evandro Chagas Institute in Belém, Brazil. At the lab, researchers thaw the insects and sort them under a microscope. These mosquitoes may be dead, but their viruses will be intact, waiting for the appropriate conditions to spring back to life. In a phase of the project that gives rich new meaning to the term the daily grind, the bugs are smashed into a liquid. That green mosquito soup is then injected into lab mice. Some will develop problems with locomotion. Some will die within hours. Some will develop infections of the scariest viruses known, diseases like yellow fever. The Institute has isolated 30 strains of virus that cause disease in humans. From mosquitoes alone, the Institute has found 40 viral strains the world never knew before. Insects are not the only way viruses travel. During the Korean War, thousands of GIs were felled by a strange fever. Later named after Korea's Hantan River, hantavirus caused hemorrhaging, kidney failure, and death. It was eventually traced back to the urine of field mice who lived in rice paddies. A similar illness appeared suddenly in the southwest United States in 1993. Disease detectives from the Centers for Disease Control quickly targeted the hantavirus group. They too traced it back to mice.
Once we had the rodent reservoir, then we had an opportunity to try to plan specific strategies so that people could avoid those particular kinds of rodents.
Avoiding the rodents is all we can do. At this point, we are still vulnerable to hantaviruses. There is no effective treatment, and outbreaks are virtually impossible to predict. Nothing shows our vulnerability more starkly than AIDS, the disease caused by HIV. It is a virus whose relentless march around the globe continues to shape the history of this century.
Robert Michael McCabe.
No one has pinpointed HIV's origin. At the Salk Institute in Southern California, Dr. Salk has turned his attention to AIDS. He accepts the idea that it was a mutation of a monkey virus that first appeared centuries ago.
It moved into the human population, very likely as a result of the ritual use of monkey blood. And then it remained at a low level of frequency, probably in villages where it was contained. And then, as civilization began to expand and travel became possible, not only in airplanes but in trucks and other forms of transportation, and people moved about, they simply spread it.
HIV, human immunodeficiency virus, is one of a fast mutating RNA family.
This HIV virus, which is really very smart, not only will go into the immune system and kill the immune system, but it doesn't do it immediately. It can remain latent for a long period of time. It uses this property of retroviruses to make a DNA copy of itself, which will go into the host, integrate there, and where it can remain silent. And then the killer cell cannot do anything, just sees the cell, it's a nice cell, cannot see that inside there is the invader is hidden. So the fact that the virus can be hidden makes it extremely difficult to fight.
The search for ways to cure AIDS brings researchers together from all over the world. They report on everything from funding to the health of prostitutes in the poorest slums of Nairobi. Discussions focus on why some people who have been exposed to HIV don't have AIDS. These women may represent the first known cases of natural immunity to HIV.
Following that evidence of an experiment in nature, which is very much like what is being observed in Kenya, it begins to suggest to us what would be the strategy that should be used in developing a vaccine. If we can begin to understand the nature of the relationship and how it functions, then we can develop strategies to cope with it. And the fact that there's clear evidence in humans that there is such a thing as a successful interaction with HIV is all that we need to know. That tells us it's possible. Nature's telling us something.
At Fort Detrick, Maryland, an Army Special Forces medical team is on 24-hour alert. If a soldier gets a disease that's deadly, infectious, unidentified, or all three, these medics are on the first C-141 out of Andrews Air Force Base. With typical military precision, they call themselves USAMRID. Located on an Army base 45 miles from Washington, D.C. Working out of a facility best known for biological warfare research, the team could be on the front line in the war against emerging viruses. They are prepared to face some of the worst diseases in the world, like Ebola or green monkey fever. Relatively new and incredibly deadly, Ebola was first identified in the Sudan and Zaire in 1976. By 1989, it threatened to infect a community just 70 miles from Usamrid. Fortunately, it was just a threat, but that's not always the case. A person unlucky enough to contract some exotic disease, known or not, could quickly find himself on his way to Maryland.
Ready? Everybody ready? We're ready. 1, 2, 3. 2, 3. Good. Got him.
It would be a strange trip, with the patient watching the action first from inside an isolation stretcher, then from an airtight capsule aboard a cargo plane. The virus, the victim, and the medics would be off to the sealed hospital rooms at the Institute. The pressurized spacesuits may make them look like miniature parade balloons, but if the suit should spring a leak, air would rush out instead of in. The medics pass through disinfectant showers to a room where the air is carefully filtered, where wastes travel through cement-encased sewer lines to be sterilized. Once the patient is in place in the hot suite, the hard work can start. In a different type of hospital room, viruses are actually being used to help rather than hurt.
I think it would be wonderful poetic justice to think that viruses, our enemies, could be used to help cure genetic diseases. I think in the future we can expect to see more and more of viruses being used in gene therapy and obviously intentionally as vectors for introducing new genes.
Researchers are in the beginning stages of using a virus's ability to break into a cell to change inborn defects in the genetic code. Experiments are underway to deliver a gene missing in people with cystic fibrosis. In this case, the part of a cold virus that makes you sick is deleted and replaced with the gene needed to cure cystic fibrosis. Then, it's set free to do what cold viruses do. It targets the cells of the respiratory tract. But instead of taking over the cell for its own purposes, it delivers the missing DNA. So the relationship between viruses and humans continues to evolve. Just as they have always used us, we are now using them. Regardless of who is in control, mankind's tiniest foe is not going away.
I think if a virus comes along with a broad enough host range, that we could suffer a real serious depredation in the number of people in our species. I think you have to consider that as a real possibility. I don't want to be a doom prophesier or say what's going to happen tomorrow or so on, but I think that HIV should have run the wake-up call.
HIV, the AIDS pandemic, I think should be a lesson to all of us, and it's sad that we have to pay such a heavy price to relearn a lesson we've known for many years, that the infectious diseases will never disappear. We can push them to the margins of our existence. We can try to control them. By and large, we can learn to live with them, but we have to learn to deal with them, and we have to deal with them. We can't just ignore them.
Viruses are part of nature. Can we not accept the fact that they're part of nature and so are we? It's all here. Do you want an exclusive right We're going to have to accept viruses. They're not going to go away.
So we stand locked in mortal combat with the humblest of Earth's creatures. And the best we can hope for is a truce. The alternative is that humankind, pleased to consider itself the pinnacle of creation, could fall victim to some drifting bits of genetic material straddling the line between life and death.